whole genome and whole exome sequencing. My understanding is down the road you'll have some other speakers who are going to focus a little bit more on particular topics. So really, hopefully today can just provide a primer to help you understand some of the technology and how it's being used. To give you a little bit of a description about my role here and what I do and what I don't do. I am a clinical genetic counselor, so I see solely pediatric patients at this time clinically. And as Leanne mentioned, I'm also part of a research study where we are offering whole exome sequencing to all of our children who are diagnosed with either brain tumors or solid tumors. So most of the work that I'm doing is clinical, so I'm not working necessarily in a lab or in the analysis or bioinformatics side of whole exome sequencing, but I'm often working with the teams at Baylor here that are doing that. I think that really highlights a point, though, about these testing technologies is the multiple amount of teams that need to go into getting the test ordered, figuring out the right patients to order it for, what goes on in the lab, and what goes on with interpretation of the results. So with that, I'm going to go ahead and get started. My hope today is just to provide you with an introduction to next generation sequencing, which is really the technology that both of these tests stem from. I am going to talk about the differences between whole exome and whole genome sequencing. Also touch upon the differences of the applications in a research setting versus a clinical setting, when you might consider ordering one of these tests in its various forms, and things to consider when you're ordering the test, so questions you might want to ask yourself, and then also future directions. So I know that we have quite a diverse group here as far as backgrounds go, but I think for everybody it's important to go back to the basics of what we're talking about.
Sorry, real fast. Are you showing your screen?
Yeah, can you not see it?
Oh, yeah, no, I can't see it yet.
Okay. Let me see.
Oh, yeah, so I have my screen up, so hold on. Let me see if I can give it to you. Sorry, guys. Let me try again. Oh, here. Maybe I have to do that. Okay.
Oh, there we go.
There, sorry. Okay. There we go.
Can you see it now?
Yep, sorry, thank you.
Oh, no problem. I wondered why I couldn't see the sidebar.
Yeah.
Okay, so just a little, going back to the basics of what we're talking about is really, I just want to emphasize the fact that a lot of the focus, or all of the focus on these tests is really sequencing. So when we think about our DNA, there's several ways that we can look at it, and sequencing is really reading through letter by letter. So there are other technologies that can pick up other types of differences in our genes or our DNA, but everything that we're going to be talking about today centers around sequencing. I thought that I would start by going back a little bit and address Sanger sequencing, which is really the technology that has been in place for quite a while now and has been the standard for sequencing to show the progression of where we are now with next-gen sequencing. For those of you who aren't interested in the lab side, you can zone out for a second, but I do want to kind of go over the details of what these different technologies use. So Sanger sequencing really kind of mimics what we do, our bodies do to copy our DNA. And so it uses a template of DNA that it wants to copy. A primer is then put in that recognizes a portion or wherever we want to start making the copies. But then these basically colored, they light up, probes will go in and attach based on a letter. All of our DNA is made up of the 4 letters that everyone's probably aware of, A, T, G, and C. And so every letter is assigned a different color. And what happens is as this sequences, we get different lengths. And when it stops, one of these probes will insert and stop that sequence from happening, but it tells us what letter is at that particular place. So by placing together all of these sequences, we're able to generate a picture like you may have seen somewhere before of what ends up being the entire sequence of the DNA. What's important to know about this is every single one of these is an individual reaction. These reactions have to take place this side. It usually takes place in kind of a well plate, and then it has to be placed into a machine that then reads all of the probes in order to generate this figure or these results. And so it is a multi-step process. So this has been being used for many years now and really is the standard. But scientists then began to try to figure out how could we make this process go a little bit faster in the hope that we can start, once we had done the whole genome project and want to begin moving this more into medicine, how could we make a faster process? So the idea of the shotgun sequencing came up, and really the title shotgun comes from the fact that we separate or blow apart the DNA into many pieces or fragments, and then we work to align them and look at overlapping areas so we can figure out the overall sequence and put these pieces in place as they should go. So this diagram shows you, focusing on these three pieces, you can see where certain letters overlap, and then over time we're able to generate a full sequence. So that's kind of the underlying idea behind next generation sequencing. It then obviously gets more complicated, and it's not important to take away all of the pieces of this diagram. I'm just going to give you a general idea of the process. I will point out that this is a review article, and if you were interested in the details of the process, this is a good review article to go to. And it does, this picture talks about Illumina. It's not any ties to Illumina other than it's a very nice diagram. So what really happens once we get these DNA fragments is they end up attaching adapters to the end of the fragments. And these adapters can then recognize a place on a template. These templates can be things like a slide or a bead, but it allows each of these fragments to attach to the template. And then the first process, which is covered in all of these pictures, is really just amplification, or increasing the number of each of these sequences that we have. And so this happens in a multi-step process. They call it bridge amplification because it forms these loops. What these loops allow it to do is to replicate that piece of the DNA without anything being washed away, because everything extra is going to be in a solution. So this continues over and over and over again until you get a group of clusters. And so this is all going to be copies of that same portion of sequence. And then that is sequenced using, similar to what we talked about in the Sanger sequencing, these probes that light up. But instead of having to do it in a two-step reaction, this can all be done at the same time. So instead of sequencing in a reaction and then placing in a machine, as it's sequencing, a machine is reading each of these probes to see what letter is put into place. So in the end, you get multiple reads of sequence that we now know which nucleotide is in each place. And then we refer to the reference genome to figure out, is that sequence that we're seeing the sequence that's similar to what we call the reference sequence, or are we seeing changes or variants in the sequence? So the reference genome has been assembled based upon the accumulation of multiple genomes that have been acquired through the Human Genome Project. And it serves as a scaffold. So this would be, each of these reads is similar to these that we saw in the previous slide. And they line up according to the scaffolding. Of course, there's room for error when this does line up, and that's areas such as where there are nucleotides that are repeating. There's GC-rich areas where we see lots of Gs and Cs. So there's different ways that this process can have error, and that's one of the things that is worked on to improve next-generation sequencing. But that is the basis of how we end up getting the sequences. We align each of these small chunks of reads until we're able to acquire a full sequence. This is what a result would look like coming out of, this is a whole exome sequencing result. And so what you're looking for, you can see here and there's small changes where things are out of place. But what you're looking for is when there's a consistent read that's different from that reference sequence. So this is actually an example of a P53 mutation, which is a cancer susceptibility gene. And once we see something like this, we know that there is a variation there that's different from what we would expect based upon the reference genome. Once these variants are identified, there is a lot that goes into the process of figuring out which of these are important and which of these are not as important. Part of this is going back to the literature or other research that has been done in the lab to see if any of these variants are actually been associated with disease already. How rare is the variant compared to what is kind of general population variation? So each of us obviously have differing DNA, and that's what makes us unique. And so some variations are common in the population, and when they're common, that makes it less likely for them to be disease-causing. But if we're seeing something that's rare, that increases our concern. Also, looking at mechanisms of inheritance. So I'll touch upon down the road here, but we do ask for parental samples. So we can see if this is something that's new in a child or in a patient, or if it's something we're seeing passed down in a certain way in the family. We can look at how that variant changes the biology of that gene. So ultimately our genes are form proteins which function in our body. So does that variation change the protein enough to cause a significant problem? So is it something that completely stops the protein from being made, or is it a missense mutation that perhaps might not be causing as much of a problem? Also, we look for the presence in other affected individuals and absence in unaffected controls. So if we see this variant in people who have a certain disease and we don't see it in, say, their relatives who aren't affected, it definitely makes us more suspicious. And have these previously been associated with the phenotype that we're seeing? So have there been other case reports of individuals who have similar features, whether it has a syndrome name to it yet or not? And then lastly, sometimes it means going to the lab and to figure out if we are trying to figure out, say, how much does that change the protein, actually doing experiments in the lab to figure that question out. So this is a new technology, and it's continually being improved. One thing that they have come up with is this concept called capture. And whole exome sequencing is actually a capture approach, and I'm going to go into more detail of that in a minute. But really what capture means is that we are looking only at a subset. So instead of looking at all of the genomic information, we're just looking at either a group of genes, or in exome sequencing, just the exome. There's also ways at improving the mapping. So if you think back to that picture I showed of the reference sequence and the different ways that those pieces can misalign, there's different technologies that go into making that less likely to happen. These aren't necessarily important to remember, but as you're doing your research on certain tests, paired end really just means that you're sequencing from both directions. Long mate pair means that you're trying to create longer sequences. One of the problems with next generation sequencing is that unlike Sanger sequencing, which has very long reads, these are very short reads, which leads to error. And so the longer your reads can be, the less error that you might see. And then strobed reading is that you're basically sequencing in one direction, but you're just kind of skipping along different places, so it can all be pieced together. So that's pretty much a summary of the technology side. And the next generation sequencing is used both for whole genome sequencing and for whole exome sequencing. And I'm going to now talk a little bit about these different approaches and their utility. So if you were snoozing during the technology part, this is when you can come back and pay attention. So your genome is basically all of the genetic content that we have. The exome is only the portions of our genome that we know actually code for protein. So it's where the genes that we actually know do something are. Approximately 180,000 exons are in the genome, and that's arranged into approximately 22,000 genes. So when you're ordering a whole exome sequencing test, you're basically ordering a test on all approximately 20,000 genes. What is important to know, though, and I'll come to at the end, is that the exome represents only about 3% of the whole genome. So there is a lot more genetic information in the genome than just the exome. So going a little bit back to biology real quick to help explain how we get genomes and exomes. If you remember back to biology, your DNA is transcribed into RNA, and it's your RNA that is actually translated to make a protein, and it's the proteins that function within our body. So during the process when the DNA is being turned into RNA, that is when we see the differentiation between what we call exons and introns. So exons, again, are the coding DNA, and the introns are everything else. So when the DNA is first copied, the introns are included. But when that DNA is turned into RNA, the introns are removed, the exons come together, and that is what's read in order to get a protein at the end. So when we talk about whole exome sequencing, we're looking specifically at the exomes. So there's one extra step that goes into the testing process, and that's what we refer to as capture. And so in different ways, again, sometimes this happens on a slide, sometimes it can happen in a solution, but they place a probe basically in of the information that they're interested in. So this, in the case of exome sequencing, would be probes that would match and allow the sequences that are involved in the exome to attach. And so whether they attach through a slide or in solution to things floating around in solution, once everything else is washed away, all that you're left with is the exome, which is the light blue here, and then the red would be all of the intronic information. So that's removed. So then you just have the pieces of the exome that are left over, and then that whole process I showed you at the beginning would start at that point, where we would amplify all of that information and then go on to sequence it. So why exome sequencing? If it only represents 3% of what's there, it's currently estimated that about 85% of disease-causing mutations are going to be identified within the exome. So right now it's also has lower cost. We're doing this experiment on a lot less material. And part of what goes into the cost as well is things like data analysis and data storage. So you can imagine when you're doing whole genome sequencing, you're going to get a lot more of those variants that are going to need to be interpreted. And likewise, there is cost that goes into storing all this information in databases. And so you have that much more information in the genome that needs to be stored. And another piece, which I'm going to go into a little detail here because it is important to know when you're ordering these tests, is something called depth of coverage. So depth of coverage really refers to how often one letter in the genome is read in all of these fragments. So for instance, if your coverage is 12 times or 12x, it means that that letter is seen in 12 of those fragments that have been produced. The higher your depth of coverage, the better your accuracy, because you can imagine if you do only have 12 reads and you have a couple variants in there, as opposed to if you have 100 reads and you're seeing that variant more consistently, it gives us better information. And given that we're looking at less information in the exome right now, cost-wise and in some other ways, it's easier to get a higher level of coverage. So one reason to use the exome clinically right now as opposed to a genome really has to do with the depth of coverage, because in some cases, even though a genome is going to show you more information as far as sequence goes, your exome might actually have a better coverage level than a whole genome would have for a certain gene, and actually would be the better test and would have a better pickup rate. So even when you're thinking about ordering whole exome sequencing, it is important to know the depth of coverage available, and especially if there's a certain gene that you're going after, it is worthwhile to call the lab and find out what their depth of coverage is on that gene or set of genes, because some areas are difficult to sequence, and so that's important to know before you order the test. So some of the variation that is detectable, and this goes back to my very first slide. So when we talk about sequencing, we're really reading through letter by letter. This is a little bit different between whole genome and whole exome sequencing. What you can pick up, you're going to pick up nucleotide changes, so you're going to pick up changes in the letters. You can pick up small areas where maybe a few letters have been inserted or deleted. And more so with whole genome sequencing, you can pick up structural variation, but still some limitations of the exome are not being able to pick up deletions or duplications or translocations or different rearrangements, which can become important, especially in things if you're looking for a tumor, which sometimes we know are caused by certain rearrangements, or you're looking in a tumor. You might not be able to pick that up with whole exome sequencing. One of the hopes of whole genome sequencing is that we're going to be able to improve that. Mosaicism, though, is one good thing about this method is that we're actually able to, especially the greater your depth of coverage is, pick up mosaicism and have a guess at how much mosaicism is present, because we can actually see how many of those reads carry a certain variant. So that's a strong suit of this test, is we can learn more about the level of mosaicism. This is just an example to illustrate, and this is with an exome test. But when we think about particular causes of Wilms tumor, there are several different syndromes that can lead to a predisposition to Wilms tumor. And if you were to order an exome sequencing test, only one of those types, which is caused by a point mutation, is going to be picked up with the current technology. So again, when you are thinking about ordering a test, it's important to think through this as far as what the limitations of the exome might be, as opposed to other testing that's available. So to kind of move across and talk about the use of this test in different settings, obviously this test really began more in a research setting and doing things like gene discovery. And so the way that the sequencing is used in a gene discovery situation is really, ideally, you're going to have a pedigree that has both affected individuals and unaffected individuals. So for those who may not be as familiar with pedigrees, these individuals who are colored in are affected, and those who are not are unaffected family members. So if you are able to get a sample from both affected individuals and unaffected individuals and do the sequencing, it really brings us to this concept of a Venn diagram, and where you're able to start comparing what we see that is different between the two individuals and what we see that overlaps between the two individuals. And of course, what's overlapping between them is what's most interesting to us. The reason then that an unaffected individual becomes important is now they can tell us other things that may overlap just because these individuals are related will be found in this region, whereas this region becomes even more important to us because these are unique to the affected individuals as opposed to just being a familial thing. So to illustrate that a little bit more, if you were to just do a sequencing test on 2 individuals, you might come up with something like 2,000 variants that end up looking important enough to look into a little bit more detail. By combining 2 affected individuals, you could bring that number down to about 900. And once you bring in a parent that isn't affected, you can bring that number down to about 200 variants, which gives you much less to look at. And so that's really the way that this test became useful in discovering some of the genes that were responsible for syndromes where we hadn't identified the cause before. I apologize if you can hear my phone ringing. So the concept then is of shared variation. So we want to look at things that are rare. We want to look for changes that alter the protein, which I kind of talked about previously, and then also things that are shared between individuals who are affected. So then thinking about this test in a clinical setting, what I just showed you is a concept called trios, and that's when we take 3 family members and we actually perform whole exome sequencing, the entire test on all of them. That is useful, especially in a situation of gene discovery, for exactly what I just showed, and that it really helps to bring down the amount of variance. In a clinical test, you're not always going to, or they aren't always going to be doing trios. In some situations, what they'll do is just be doing whole exome sequencing on the child, and then going back. They do Sanger sequencing on any variants that they find, typically in the child, and that's really just because Sanger sequencing is the gold standard at this point. And then they would go back and just do Sanger sequencing on those specific variants that were identified in the child, as opposed to doing the whole test. Another thing that's important, especially for clinicians and genetic counselors to know with whole exome sequencing, is it is strongly driven by the clinical and family history information that you provide. I know we've all filled out forms many times that have asked for clinical information, and it is important to know that this really does drive what they're doing in the lab, because they're going to be getting so many variants that are going to push out of this test that they're using that clinical information to zone in on what might be important. So it is important when you're filling out these forms to give as much information as you can. Sometimes I actually will even include a report with samples just in case I don't feel I can summarize it within the requisition that they give you. It also is important when you're looking at different labs that are offering the test to look at the difference between their consent and what they're going to be reporting back, because this is going to vary, and this also varies, differently between research and clinical, because certainly with research sometimes you're not even getting results back. But different labs do have different consent forms right now as far as what you can opt in or out of, or what you're just going to get. And I'll talk a little bit about what the report information you might get back on the report would be in a minute. And then it's also important that these results go in the medical record. There is some concern with patients when we have been ordering this test, because now there is so much genetic information that's included. This goes into the medical record. What does that do as far as things with genetic discrimination? So that is an important piece to keep in mind, and something that's a little bit different of this test from our previous. Obviously, we were still putting genetic information into medical records, but this is a lot of genetic information, and so that raises concern for some patients. So what information might be reported back to you? And again, this can vary by labs and the different tests that they offer. But what you might get back is, of course, what we're looking for. So disease-causing mutations that are related to the reason that you ordered the test. So that's going to be a positive test result, and that's really what we're looking for when we're ordering the exome. You might also get back, though, variants of unknown significance that are related to the phenotype. And this is, I think, what a lot of us are nervous about, how we're going to explain these variants. And that is going to be a challenge of this test. Typically right now, I explain it to families that these are changes. I explain how they're not necessarily associated with what we see going on right now, but the reason they're in the report is if things do change over time, that we can go back to the report, or if we learn more about a gene in the future, we can go back to the report. And in general, I've found thus far that families have, some have more questions than others, but many people are able to accept that information and not worry too much about it. And the next point is, I think, an area of great debate amongst the genetic community right now as far as what are considered medically actionable mutations. There's ACMG recommendations that have come out that have talked about what needs to be reported, what needs or must be reported. Different labs are handling this in different ways. But this is really kind of changing the paradigm of genetic testing, I think, for us, because now we are, we could, in testing a child, come up with adult onset cancer condition mutation, and historically that's not something we've dealt with in the past or done. So it brings out many questions, which I'm sure will be talked about in your ethical lecture, ethics lecture. There's also the choice to get carrier status often. So that is offered to families, and again, something we usually haven't done in children, but can provide information for reproductive decision-making for the parents, even in that situation. Pharmacogenetics. Usually the pharmacogenetics that are being offered on these panels are a small subgroup of medications. They're ones that FDA has shown that there's some kind of association or utility, but those are available on some of the exomes. And some also offer mitochondrial DNA mutations, which can be helpful. And I do want to point out, as I'm talking about this, most of what I'm talking about is whole exome sequencing, because in general, that's really what's being ordered more so on a regular basis with in the clinical setting. So when might you consider ordering one of these tests? And it's important to kind of start off that it seems early results are suggesting that about 20 to 25% of patients are receiving a diagnosis, even if they've had a previous set of genetic tests. But some of the patients you might think about this test in are that diagnostic odyssey that we all have heard so much about, but really these kids where they have so much going on and we can't quite figure it out, they are a good candidate for the test. Also, it can become useful if you have a condition in which multiple genes are involved. Here at the hospital, we've actually ordered the exomes. Individuals with Charcot-Marie-Tooth can have certain reactions to chemotherapies. So when we see children who have similar reactions and then a concern is raised for Charcot-Marie-Tooth, we actually have ordered exomes. But this ends up being a money thing and also just a coverage thing, that this test sometimes ends up being a better way to go to try to look at all these genes at one time, going after one condition. And then also those cases where in the past we would have ordered tier testing. So we may start with one gene. If that comes back negative, then we might think about the next. And in this case, this really allows us to look at all of them at once. Another situation that you may or may not have thought about is if there's a gene of interest that's not yet clinically available as a test on its own. So, there's certainly genes that have been reported that are associated with certain conditions, but maybe a lab is not offering that test yet. That gene can be looked at in the exome. So those are all situations that you might consider doing an exome test. So the limitations of the test, timing is probably the biggest one. And so most of these tests you're looking at least a 3-month turnaround time. And it really is a limitation. And it's because so many steps need to go into, even sending the sample out, receiving, processing, prepping the sample before it's even ready for the test, going through capture, going through the sequencing process. And then once all that's done and you actually have the data, there is a lot that goes into analysis from that point forward. There's people in the labs who are searching databases, searching literature to really make sure that they're not missing anything and that they're reporting everything of importance back. So that's something I think every lab is out there trying to improve, but it's what we're looking at right now. So in some cases, it's probably better, at least if you have even if it's a situation maybe where you're thinking about tiered testing, but that first test is important time-wise, you might just go ahead and order it. For instance, here with some of our patients who may already be enrolled in the study, we know they're getting whole exome sequencing, but say they have a very strong history for P53 or something like that, and there might be a medical need to know that answer right away, then we would just go ahead and get the test done that has a quicker turnaround time. So that can play into the clinical setting. Also, that there are types of genetic variations that are missed. So like I said, right now, with exome sequencing, we're not able to pick up deletions and duplications like MLPA might. And so that is, you need to really ask what you're looking for, know if there, is a gene that you're going after, to know all the ways or all the types of mutations that might occur in that gene, so you know if it, how useful the exome would be to you. Certainly, there's labs that specialize in certain genes or diseases that actually might be preferred if that's something that you're looking at, because they're going to look at that gene more thoroughly. An example of this for us is retinoblastoma. We send our retinoblastoma patients to a lab in Toronto that has been studying that gene for years, solely that gene, for a while. And so they've just looked at it in so many more ways than the exome can. And then there are several gene panels that are available right now, and I think some people hit that question of, well, if I'm going to order a panel, why not just order the exome and do the whole thing? And there are situations where the panel might be a better test for the same reason. Some panels do include, deletion duplication analysis for some of the cancer panels. So if it is more of a subset or a specialized thing that you're going after and there's a panel for it, it might be appropriate to do the panel instead of the exome. And so those are all things to consider as you're deciding whether or not to do the test. So I think I've kind of touched upon these through most of my conversation, but important to know, clinical information is important. It guides the analysis. Know the depth of coverage of, especially if you have a gene of interest. It's worth the call to see how well covered that gene is. Is the test doing a trio approach? Are they doing Sanger confirmation on the parents? That's just important to know, even sometimes to describe to the parents. Carrier status is an example of this. If you were to find out, some labs might report back which parent is a carrier of what, but if parent A carries the same variant as the child, it does not mean that parent B does not carry a change in that gene because they didn't do sequencing of that whole gene, they just looked at the variant. Look at your consent forms so you know what information that you're going to be getting back, and it's best to consent the family and make them fully understand, or have them fully, I shouldn't say make, have them fully understand what information they are going to have back, and then know what information is going to be returned to you. So are you going to receive parental results? Is everything on the test Sanger confirmed? Because some changes or variants that might be picked up may not go through Sanger confirmation. And then ask yourself, is there another panel or another test out there that might be more appropriate? So some practical implications. One here, for example, is that we use institutional billing. So parental samples are technically free as far as the lab goes, but to actually get a parent poked and have that sample shipped, they do sometimes have to pay a fee for that. So how are you going to get the parent samples? How are you going to set up your consent and disclosure visits? Is it, stepwise approach? Will you do the consent all as one appointment or with a clinical visit? So those are decisions to make. And then, of course, insurance coverage. It depends on how your institution does it. If you have institutional billing, if you are submitting it yourself, a lot of companies will help you with that process. I can't say that there's rhyme or reason right now to anything I've seen with insurance coverage, but it does get covered. But I've also had families where it's been denied, and I've had families who've paid a chunk of money to get it done because it was only partially covered. So the ethical issues, there's many, and this is going to be a talk and should be. So, you know, the process of the variants of unknown significance, how we give those back, how we describe them to families, what are we going to do with these medical actionable mutations, and all the things such as identification of adult onset disorders that can come up, certainly privacy issues with genetic testing, and then we often do test parents in a genetic setting, but not always. And this is a test where they ask for parental samples, so certainly things like non-paternity can come up. And so the future, really, I think what the future is looking at is how they can continue to improve the testing technologies and get the turnaround times down. The utility of whole genome sequencing. So some of the promises of whole genome first are, you know, the things in the genome that we don't necessarily know that much about yet. Could we learn more? There is a little bit more possibility of understanding the structural variation differences using the genome. So those are just some examples of the way that that can be used. And then, just as I mentioned, improving the pickup of other things like copy number and structural rearrangements. And this was an article that came out in the New York Times and with the mystifying name of DNA dark matter. But really, I think that there is a lot still to be explored what's going on outside of the exons. Right now, as a clinical test, the exon sequencing test is a great one. It can do a lot for us, but we still have plenty to learn, and we might be having a similar talk focused more on whole genome sequencing a few years out. So just to very briefly go, because I want to wrap up for questions, this is the study that we're doing here at Baylor. It's called Basic 3, where we are offering whole genome, or whole exome, sequencing to all of our kids diagnosed with brain tumors and solid tumors. That's regardless of family history. So even if a family wouldn't be being referred to the clinical or cancer genetics clinic, they still are offered the test. But the interesting part of the test is we're looking to enroll about 280 children. We are doing both blood and tumor. These reports, it is a clinical test that goes into their medical records. That test result is given to them by their oncologist, and that is with us. There's 2 genetic counselors on the study, so the oncologist does have a genetic counselor in the room with them while they receive the results. But we're looking at things like medically, how useful is this test? What's the utility? Down the road, if these kids relapse, is anything we learn, especially from things like the tumor, going to redirect our care? There are additional research studies being done in non-clinical labs to look at other technologies, such as whole genome sequencing. And then we have 3 projects. The first is the clinical team, so that includes the genetic counselors, the oncologists, and this is giving results to the family. The second is those people who are working in the lab to analyze these variants, to produce the reports. And then the third part is the ethical side of the team. So we're looking at our disclosure visits are audio recorded. So we're looking at physician and parent communication, how these things are described, what language is being used, what questions the parents are asking. We are looking at how the oncologists see this as useful or non-useful, and also we're interviewing the families to see what they're getting out of it. So it is a very interesting project to be a part of, and it has allowed me to have a lot of exposure to this test, which has been helpful. So I just want to acknowledge Dr. Plon and Dr. Parsons are the two PIs on my study. Dr. Sharon Plon is who I do see patients with, and she is fantastic at what she does. Bradford is a genius that works in our lab who helps me understand the technologies and help me with this. And then our Basic 3 team and our oncologists. And then I also want to, of course, thank the Hawaii Department of Health and the group there, just because they raised me as a little genetic counselor and sent me on my way to do these things. And I am very, very thankful for my ties to the group. So with that, I will open up for questions.
Thank you so much, Sarah. That was a great presentation. Everybody is still on mute, so if you do have questions, please type them into either the chat box or the questions box. And Sarah, I do already have 2 questions for you.
Okay.
So the first is, how many participants are enrolled in your study to date?
I think we are, sorry, I'm anal. I need to be on a pretty slide to sit here and talk. We are over 80. So we were just discussing what we were going to do when we hit our 100th. So I think we're in the 80s right now.
Okay. And then the second question is, when whole exome sequencing results are reported out, do you know, will the lab specify the depth of coverage, or is that something that we need to call the lab?
I would actually have to look at a report to see if it's in the technical language, but it's nothing that is obvious. So that's something I think when you're researching labs, that is information that you can obtain, but it's not something that's very obvious on the report.
Okay. Okay. And then I had a quick question. Since you are involved with figuring out, family preferences for receiving data, how have you found families' understanding of this testing? Do they get it? Do they appreciate the nuances? What has been your experience with families and this test?
So and a little bit different, and it has been an adjustment for me, especially coming from more the public health side, who would be doing the interviewing and that part. So I'm not part of the Project 3 team, so I'm not involved in the interviewing the families, but my role with them is actually in the disclosure session, so giving them the results and interacting with them, which is still, I think, telling. But honestly, I've been surprised at how well the families do receive it. I think people get it. At least for those that have enrolled, we have a great research coordinator who consents them, and we really worked on that consent process thoroughly to make sure that they knew what would be coming. But of course, you have some that they kind of glaze it over, and they just hear the bottom line of whether it told them something or it didn't tell them something. But families are very excited about this test. We have, I'm not even sure the number right now, but majority of them say yes to doing it. And I think some are actually disappointed when they don't find something out in the end. It's a little different here because we are doing it on the tumor. So I think a lot of families are really hoping to find something in the tumor that tells them useful information about treatment. But in general, I think that I was more worried than I needed to be, I feel like.
That's good to know. Are there any other questions for Sarah? So we have been recording this presentation, and I will make this available with Pauline's help on the password protected section of our website, hopefully within the next week or 2. And Sarah, if people do have other questions in the future, since I'm not seeing any popping up right now, can they email you, or can I have them email you?
Of course, you can send. I'm sorry, I could have put it on the slide, but you can send my email out. That's no problem at all. I'm more than happy to.
Great. So one more chance, going once, going twice. Anybody else with any questions for Sarah while we have her as a captive? Well, Sarah, thank you so much for this presentation. It really helped me. I didn't really know very much about these techniques. And so it really helped me understand both whole exome and whole genome sequencing so much better. And for everybody else, we are having, this is a series, so the next webinar is going to be about clinical applications of whole exome and whole genome sequencing, and that's going to be on Thursday, August 15th at 3 p.m. Pacific time. You can find a link to join that webinar on our website, so if you want to register for that, and Sarah, you're welcome to do that as well if you're interested.
Yeah, okay.
Yeah, so again, thank you so much, Sarah, and thank you to everybody who participated, and we'll put this up on our website as soon as we can.
Okay, thank you guys.
Okay. Thank you.
Yep.
Okay, bye.